So good afternoon, and good afternoon also to everyone on Zoom. So welcome to the UCSF Academic Senate's 11th Annual Faculty Research Lectureship in Translational Science. I'm Su Guo. I'm a professor in the Bioengineering and Therapeutic Science Department of the School of Pharmacy, and I'm also a member of the UCSF Academic Senate's Committee on Research. I will be co-hosting this lecture with the chair of the Academic Senate's Committee on Research, Penny Brennan. Here she is. Oh, you are on mute, Penny.
Hi, I'm Penny Brennan, professor at the UCSF Institute for Health and Aging and chair of the Academic Senate Committee on Research. The Academic Senate Committee on Research is hosting this meeting. The faculty research lecture began at UCSF in 1957 to recognize and celebrate the outstanding career achievements of UCSF research faculty. Nominations are made by UCSF faculty who consider the scientific contributions of their peers and mentors and submit nominations for this award to the Academic Senate Committee on Research, who then awards the recipients. I'd like to thank the members of the Academic Senate Committee on Research for their hard work reviewing the many outstanding applications we received. Please note that the nominations for next year's faculty research lecture are now open through June 10th. You can submit nominations online through the Academic Senate Service Portal. Please consider submitting a nomination to one of the three categories: clinical and population sciences, basic science, and translational science. Please note that the clinical and population science category awards researchers who are doing work in the clinical fields, as well as social and behavioral sciences and policy-related fields. Please note also that the Committee on Research welcomes and encourages nominations of faculty from historically excluded groups or those underrepresented in medicine and science. Sue?
Great, yeah, thanks, Penny. So today we're proud to honor our colleague, Dr. Diane Barber. Dr. Barber is the endowed professor and chair of the UCSF School of Dentistry's Department of Cell and Tissue Biology. So there will be a chance for questions and comments after the lecture. And also any questions and comments from the Zoom can be made using the Q&A functions. And Penny will moderate that Q&A. And I will be taking your questions here in person. So next, I would like to introduce Francesca Aloisio. Great. So she's a postdoc in Diane's lab, and also is a member of Barber's lab. And then you're gonna have a video to show us, right, to introduce Diane. Yeah. And then you're gonna introduce.
All right. So beyond Diane's lifelong commitment to scientific excellence, she's also played an immeasurable role in forming the lives and careers of dozens of trainees throughout her career. And so a couple of us current members of her lab wanted to also highlight this essential aspect of her career as well.
Hi, Diane. Greetings from West Virginia, from Brad, Katie, and Annabelle. Congratulations on the Faculty Research Lecture Award. It is well deserved.
Hey, Diane. I'm very happy for this award and for your outstanding scientific career.
Congratulations, Diane. We caught up with a few of your former trainees for a little walk down memory lane throughout your amazing career here at UCSF.
I was still in middle school, and I was so inspired by you, and you became my role model in science.
I met with her to discuss the cell bio exam. Instead, she spent the entire hour convincing me to rotate in her lab.
And I just clearly remember thinking, like, wow, this is a really cool person, a cool scientist, and someone that I can tell would be a great mentor whether or not I join her lab.
I just thought, she's so cool. It was a total no-brainer to join her lab, because I kind of want to be her when I grow up.
Life is short, so always ask important questions.
I am an oral and maxillofacial surgery resident, and I still use a lot of what she's taught me here in my clinical research.
To make the choices that allow you the most options down the road, or make the choice that leaves the most doors open for you.
I think my favorite Diane catchphrase has to be, of course, resilience.
To be resilient, but also how much fun a career in the sciences can be.
The new discoveries can be made through collaborations with experts in various fields.
It's okay not to get the result you thought, because it's called research for a reason.
The best way to be an outstanding leader, to set a strong example for your trainees. I always strive to match her work ethic.
Is to always be fearless when approaching a new problem or a new experiment that I haven't done before.
My favorite Dianism? What's the question? I mean, this is the answer to the question.
What is the question?
What is your question?
But what is the question?
But what's your question? And I still use that all the time in my own lab.
And of course, the infamous, but what's the question?
One of my favorite Dianisms is when she calls PFK the poster child of allosteric regulation, that I found myself saying that to my students when I taught metabolism earlier this year.
Favorite Dianisms, it's not a pump. Don't ever call it a pump, it's an exchanger. Other favorite Dianism, about Pokemon Go. It gives me the heebie-jeebies.
Probably when she would always ask me, is the research fun? And I think that positively reinforced to be excited about my own research and my own work.
The researches at the Barber lab has always been fun and creative.
It's just how to have fun doing science, and Diane is often in the lab, and her enthusiasm is just so infectious, and it makes for such a great work environment, and it makes her such a great role model and mentor.
When she gave a talk at ASCB, and what first struck me was how far-reaching her research was, how her research was focused on questions, and questions that were essential for life, essential for understanding disease, and essential for understanding integration of biology. And I remember leaving that seminar just buzzing with energy, and I felt like my brain was on fire with the possibilities of pH and biology. And that was really a life-changing scientific moment for me, and it changed the trajectory of my whole career, and I'm really grateful for it.
Thank you, Diane.
All right, so next I will introduce Dr. Sarah Knox, who's a faculty member in the Department of Cell and Tissue Biology and who nominated Diane for this award.
Thank you. That's hard to, I don't know what I'm going to do now after that. But how do we move forward? We just do that. Great. Okay. So I'm going to go into a little bit of, not too many details about Diane, but just give you an overview of who she is. And so let's start here. So Diane was born, of course, in raised in Central Valley in California. And so, that's not me. No, that's not you. But it reminded me of you. So, oh my goodness, yes, let's do this. So she was born in California, and a couple of years later, she finished her PhD. And that was in anatomy at the Center for Ulcer Research at UCLA. I'm not quite sure if we're still into ulcers like we used to be, but that was very big at that very point in time in 1985. Then she did a 2-year postdoc at UMass Medical Center, got an F-32 to fund that. And then somehow, 1985, 1987, she becomes a faculty member at Yale, right? And starts exploring the intracellular pH, and does a lot of collaborative work with experts in epithelial transport biology. What was very interesting to me, I'm not sure if everyone's aware of this, I study the peripheral nervous system and the interaction with glandular organs and glandular cells. And what I found was these. So Diane also did a little bit of neuroscience, and she got published in Neuroscience itself and Neuroscience Letters on, guess what, the peripheral nervous system and glandular tissue. There you go. So Diane let's collaborate sometime on that one. Maybe it was 1989, but hey, what's 30 years? Or is it more than 30 years? Anyway, so she joins UCSF in 1992 and establishes a wonderful lab, very young, very amazing. Obviously, maybe not these people in it, but you'll see that Diane's always been a very family-oriented person, and some of her daughters are in this image. Some of her daughters. She has 2 daughters. I think they're here, right? Thank you. Thank you for having a mother like Diane. So that was the beginning in 1992, and she has gone into a whole range of mechanisms, signal transduction, GTPases, how changes in the intracellular pH regulate cell behavior. Some of those things over the last 30 years. She's also mentored a large group of people and essentially tormented them in sport. So she's been very strong advocate of baseball, and so they had to play it, or you had to go and see it. The other thing, of course, you had to be taught how to have a good time. Diane did an exceptional job at that. So Diane is an outstanding mentor. She not only mentors trainees, she also mentors faculty. And she's won numerous awards, including these 2 here. So in 2010, Cell and Tissue Biology gets a new chair. It's Diane Barber. And she starts by recruiting people. She recruits 9 people over this time, over the last 12 years, and that includes me. So there I am with Diane. We're at this is Trump era politics. We're out there supporting science because science is like magic, but real. So she did a fantastic job at really establishing a force in cell biology, developmental biology, regenerative medicine, cancer in our department. So thank you so much for doing that, Diane. She's also, of course, she does the chair work, she does the mentoring, and she's an outstanding scientist. She's also obviously an outstanding interpretive dancer, as you can quite clearly see that highlighted in the red. This outstanding research has been obviously led to her being internationally recognized for her exceptional work pioneering studies, bridging protein dynamics and electrostatics with cell biology. This has been highlighted by her election to become an AAS fellow. And of course, she is a keynote speaker just about everywhere, internationally and nationally. And of course, I think you've already heard this word, resilience. Diane's got to be one of the most resilient people that I've ever met. She figures out how to deal with the difficulties in a very quick time, and she teaches you how to deal with them too, and she's taught her mentees how to do it as well. So thank you, Diane, for that. Fantastic that you won this award. You're absolutely deserving of it. And so Diane's now going to come up and give us a talk, and it's a road less traveled, that's for sure. A fascinating and fulfilling journey with pH. Thank you.
. Thank you, Sarah. Thank you all for joining us in person and for those who are joining us remotely. And I apologize for my raspy voice. I've got a really nasty cold. First one, I think, since pre-pandemic, which makes me realize how good masks are for not just keeping out COVID virus, but other viruses too. And I have with me the good luck notes of my granddaughter who is sitting here with blue headphones on, who drew me good luck notes this morning. Now, can you hear me okay if I'm fabulous? So I want to start out with a couple of thank yous. First of all, to the Academic Senate for bestowing me with this fabulous award, highly appreciated. Second is to Sarah Knox and Stuart Gansky, who nominated me for this award, as well as a number of people, including Todd Neistel, who wrote supporting letters. Third is to you, joining us today, both in person and for those of you who I can't see, joining us remotely. And finally is a call out to Cambria Chalfried, who is sitting up here in the front row, for this dynamite image of a zebrafish embryo expressing an intracellular pH biosensor. Whoops. And this is the yolk sac of the embryo, and this is part of her thesis project on intracellular pH dynamics and neural crust development. So as just a very brief outline of my talk today, I'm going to actually start where most seminars end, and that is with acknowledgements, because it is the people and the collaborations and the experimental models that have brought me to this point today. I was asked to incorporate my journey, both career as well as personal. And then I'll touch on a few research highlights. I'll first give a brief background or overview of our research program. And then I want to talk about two of our current projects that I believe are really innovative as well as broadly significant. And that is a new idea on how pH regulates transcription factor binding to gene promoters, and a second on the role of pH dynamics in charge changing mutations, particularly in cancer. And then, as this is a translational award, I'll end up with some translational perspectives of our work. So first and foremost is a shout out to all the trainees that I have had the incredible pleasure to guide along in their career. About not quite to, I guess a little over a half have gone on to transition into become university faculty and group leaders. And about a third have gone on and transitioned to scientists in biotech or group directors in biotech, and the rest with a myriad of different careers in science education, publishing, policy, and healthcare. So as I said, a little over half have gone on to faculty positions at institutions all around the world, as shown here, and also biotech. So this is one of my favorite quotes from a trainee. I didn't realize scientists could be so fun. And this was told to me by Mario Esquivel, who at the time was a San Francisco high school student and part of the Science and Education Partnership Program. And Mario went on and completed his undergrad at USC and then went on to graduate school. So it was a really successful step, was a very successful STEP student. But when I met with Mario at the end, and we're still in contact, and this was maybe 10 years ago, I asked him, you know, what was really most surprising to him? What did he really learn? And he told me a couple of science things he learned. He did a Drosophila screen at the time, a suppressor screen. But he said the biggest thing that he learned is that scientists could be so much fun. And indeed, we make sure we have a lot of fun, whether many different BMS themed happy hours, our annual canoe trips down the Russian River that end up with a barbecue at our home in Mill Valley. And anyone who knows me knows that baseball is a big part of our fun. Talking about baseball, watching baseball, playing baseball. And I think at one time or another, there's probably devotees of about 5 or 6 different teams in both the American and the National League in our lab. And it's been a lot of fun. So my collaborators, a call out, all 3 of these are here. Matt Jacobson, who's hiding in the back there, who we've had, I don't know, Matt, maybe 15 or more years of an incredible collaboration. We've published, I think, more than 10 publications, papers together, shared multiple grants, and really got us into bridging protein electrostatics and protein structure with cell biology. Todd Neistel, who's also sitting in the back, really got us in a new direction about, 5-6 years ago, or a little bit more, in developmental and stem cell biology. And a huge shout out to Torsten Whitman, who taught us everything we know and use about quantitative live cell imaging, and has really held our feet to the fire in terms of using quantitative approaches for cell biology. And then there are a number of others, some who are here, that we've either published with at UCSF, collaborated with, shared grants with. And I would be remiss if I didn't acknowledge here and there that this is an entirely an XY genotype. And I don't know what to say about that, but
We should have a collaboration.
it's what?
I'll collaborate with you anytime.
Okay. But I'm going to come back. I'm going to come back to this because it is a concern. It is a concern. Okay, I also have to give a shout out to all the wonderful model organisms we've used, and I think we've been pretty diverse. Our mainstay are clonal normal and human cancer cells. With Todd, we began 5 or 6 years ago working with embryonic and stem cells and iPS cells and adult stem cells, particularly intestinal organoids. And then we've used a number of different animals. Probably the first foray, quite a few years ago, was with Dictyostelium, which I still hold a fondness to. And Dicty was very instrumental in us really shoring down that pH dynamics plays a critical role in motility, or more specifically directed cell migration. So on the top, let's see, here we go. Okay, so we come in with a pipette that is releasing cyclic AMP, which is a chemoattractant, and you can see the cells now polarized. They actually execute U-turns. They don't turn their fronts into the back, their back into backs. They're highly polarized, and then chemotax towards this. And we were able to show using a pH biosensor that there was a pH gradient and an increase in pH during this chemotactic process. So we asked, what's the importance of that? And so we did a genetic knockout of a sodium proton exchanger, which is an acid extruder. And in these cells, there is no increase in intracellular pH. And you can see that the cells sort of are still motile, they wiggle around, but they do not chemotax toward the chemoattractant. They've lost directionality, they've lost polarity, and we can rescue that now if we re-express a wild-type sodium proton exchanger. We've used Drosophila quite a bit. I was not raised in Drosophila genetics. Todd has helped a lot, particularly with our work in pH and cancer. And these images in the middle are from an Eli publication from a number of years ago. And we generated a Drosophila line expressing a pH biosensor. Thank you, Joey. A pH biosensor. And when we express an activated oncogene that caused dysplasia in the eye epithelium, it also, we could see, had an increase in intracellular pH. So we asked a very simple question, well, what if we don't, what about in the absence of an oncogene? What if we just increase pH in the absence of an oncogene? And so we did that by overexpressing a sodium proton exchanger, and that in the absence of an oncogene was sufficient to induce dysplasia. And this was sort of set us on the road that I'll talk about later in terms of how this is happening. And then, as I showed you in the first slide, Cambria Chow-Fried is now using zebrafish to look at the role of pH in neural crest development. Funding, extramural, has predominantly been from the NIH over the years, both through GM and NCI, as well as in the past. And currently, Todd and I share an NSF award, and Matt and I were recently received another NSF award. Intramural awards from UCSF have been absolutely instrumental in getting us started on pilot projects and new ideas, whether it be through RAP, PBBR, or Catalyst Award. Absolutely incredible. It's just, I can't emphasize what a phenomenal resource these intramural funds have been for us. Okay, the journey. So I was born and raised in the Central Valley of California in a very large Armenian family. My grandfather's last name was actually Barbarian, and it was shortened to Barber when he immigrated here. And here I am as a cowgirl with my brother and some of my first cousins. And when I say large, I mean large. So just on my mother's side, we were 12 first cousins. And on my father's side, we were another 12 first cousins. And we spent holidays together, weekends on our grandparents' grape vineyards. It really was a wonderful life in the 50s and 60s. And on both sides, my mother and father's side, these cousins are still remain very, very close, and consequently also are quite larger. This was taken last fall. These are these same 12 cousins now with their children and grandchildren. And on my father's side, again, we get together once a year. These are the same 12. These are the 12 cousins with their children and grandchildren. Now, some of you might look at this and say, that's a real problem of population explosion from two sets of grandparents. I look at it and say, oh my God, that's a lot of Christmas cards to send. So, but it really is fun. And it's been an incredible extended family. So Leo and Helen, my parents, are first generation Armenian. My grandparents immigrated here from the Anatolia region of Eastern Turkey. In between the two massacres, most of you are familiar with the 1915 Armenian genocide, but there was also one in the 1890s. So my grandparents came here around 1910. My parents were children of poor grape farmers, and unfortunately also came of age during the Great Depression. So they were not able to go to college. But just within 2 generations, we have 3 university faculty in our family, not only me, but my brother, who is a vet radiologist and recently retired from Virginia Tech. He was a department chair. And my niece, Leah Ciccarelli, shown here as a shy child with my sister and me, is a professor in the Department of Communications at University of Washington. And she has a very interesting research subject, and that is the rhetoric of science. And she's written 2 wonderful books that I'll shamelessly plug. One is on rhetorical strategies that were successfully used by Dobzhansky and Schrödinger, but not so much by Wilson. So how rhetoric can be used successfully or not. And also a more recent one, these are both available on Amazon, on constraints of the metaphor of using science in terms of a frontier, and really argues against using that. And I think, Leah, I think you're listening. So I plugged your two of your books. Okay, quickly, the journey, as Sarah sort of highlighted for you, from the Central Valley. I did my undergrad and master's at Davis. I went to UCLA, where I received my PhD, and drove across country with my husband in an old beat up Volkswagen Rabbit, and did a 2-year postdoc at UMass Medical Center, although we lived in Boston, and then made a short move to New Haven. I was on the faculty at Yale, and as Sarah said, this is where I started studying intracellular pH dynamics through collaborations in their physiology department, which is world renowned for their expertise in epithelial transport, as Rob knows. And then after 4 years, I really wanted to move back to California. And so we took this green line. Now, this blue line, I said, we drove across country in an old beat up Volkswagen Rabbit. We had a wonderful German Shepherd, huge German Shepherd in the back seat, and a cat. And when we came back on this green line, we had the same dog, a different cat, a Subaru station wagon, and 2 kids. So it was a little bit different going and coming. So the current journey, as Sarah said, I'm currently the chair of cell and tissue biology, although I'm stepping down in 2 months, and Jeff Bush will be the new chair of CTB. Condolences or congratulations, I'm not sure which. We are a roughly 13 faculty with various research programs, about 30 students, and about 55 postdoctoral fellows. So outside of UCSF, I have 3 loves. One is, for many years, I've been integrally involved in Women in Cell Biology, which is a committee within the ASCB, and I have served as a past chair. This is an advocacy group for gender equity and diversity in sciences and cell biology. I, with Haig got me involved in teaching an annual week-long cell biology course in Yerevan, Armenia, that has been absolutely incredible. And I want to give one little call out here is a young woman. This is, I think, the first year we taught, and I didn't know her name, but she really made an impression on me because she was the only high school student that has ever taken our course. They've always been graduate students and undergraduates and graduate students, a couple of postdocs. Well, fast forward to today, and this woman happens to be with us. Anush, could you stand up? I want to give, she is just, your story is incredible. Anush was raised right outside of Yerevan, and she traveled to Yerevan to take this course in 2017, I think. And now she is an international student at University of Richmond and has come to work in our lab for a summer research fellowship. And it's just full turnaround, and it's just such a pleasure. And your story is quite inspirational. And then my husband and I have both, weekly, we coach Special Olympic swimming, weekly practices, and then we have about 2 or 3 meets that we organize every year. And we've been doing that for a long time. Okay, my 2 daughters, who are both here, Claire and Lauren, were actually 4 and 1 when I first came to UCSF. It was a challenging time, needless to say. But watching them grow and develop into incredible young women has been the best journey ever. Every summer we would go someplace all over the world. That was our big thing. We took big summer vacations. Here's a picture of us. I think you were in high school at the time, maybe. We did a surfing road trip down the coast of Portugal. And when Claire was young, she used to like to come into the lab with me to do what she called experiences for experiments. And here is Claire now at University of Washington, where she did her undergraduate and her graduate degree, collecting oysters for her own experiences, because her graduate thesis was on epigenetics of the oyster genome as a function of changing ocean conditions, temperature, acidity, etc. And she is now a bioinformatics analyst at TwinStrand, which is it a startup that specializes in duplex sequencing. Lauren completed her undergraduate degree at UCLA. And during that time, she did a semester abroad at University of Cape Town in South Africa, which was great because we got to go visit her. And then she spent quite a bit of time traveling with friends throughout South America. And then she returned to California, went to law school at UC Irvine, and she's now an attorney with the LA Public Defender's Office. Richard Ellis has been truly an exceptional partner. Here he is with the girls at the top of Aspen Mountain. Here he is with Claire dancing at her wedding, which was held on University of Washington campus. And Richard has had his own challenging career. He is a criminal defense attorney who specializes in capital habeas, and that means that all of his clients are on death row. And it's not a profession with a whole lot of success, but it happens occasionally. This is from the headlines of the Marin IJ paper from quite a few years ago, in which he got I can't remember, a stay or something. But just in the last 6 months, he's gotten stays of execution for 2 of his clients. One was just a couple of weeks ago, and you may have read about it. It was a woman in Texas, Melissa Liccio. And so that was Richard's client. And it's quite incredible. In fact, they're getting her a new trial. And unfortunately, Richard couldn't be with us today. Many of you know he just returned home after being in the hospital for a month. He had heart surgery and is recovering slowly. But Richard, I hope you're watching us remotely. And then thanks to Claire and her husband, Aaron. Hi, Bennett. Bennett and Layla are with us. Layla. Nope. So Richard and I are the besotted grandparents of Layla and Bennett, who unfortunately live in Seattle, but thanks to Zoom and Skype, we do lots of puppet shows and reading stories together. Okay, those of you who know me know that I've always been a proponent of the benefits of an academic career for time flexibility. You know, the time flexibility that I had allowed me to go to all their sporting events, work in their classes. You know, I'd come home and stay up till 2 o'clock in the morning, and I was chronically sleep deprived for the first 10 years, as most people who could see me at a 4 o'clock BMS seminar sleeping would know. It wasn't easy. It's challenging, but it was wonderful. And in terms of working in the classroom, I think my favorite thing we did is for quite a few years, Mike McMaster, shown here, and Susan Fisher, shown here, and I, when our children, each of us had 2 children, and when they were in 5th grade, Lauren, do you remember this? We organized a field trip for their classes to come to UCSF and learn about the human body. And we created spaces or stations for a hands-on, and we got everyone involved. So this is a picture of Allison Buchan, who's a longtime dear friend. At the time, she was a professor at University of British Columbia in Vancouver, and she was doing a sabbatical that year with our lab, and we got her involved teaching about peristalsis. So that was just so much fun that we did every year for quite a few years. But, you know, it has been a challenge. Raising a family, raising children, teaching, research, and service, and navigating the academic life is not for the faint of heart. I wouldn't change it for anything. 30 years later, I still love it. And I'm incredibly fortunate to be able to say that. But it's not easy. And in the last couple of years, I've written several articles on increasing the representation of women in basic science in particular, and how to plug the leaky pipeline. And three highlights that I want to make are it's absolutely critical to maintain an environment where women believe that their voice is valued and their talents. To ensure effective mentoring, both vertical and longitudinal, peer mentoring is critical. And an awareness towards achieving resilience. So I want to go back to this first, maintaining an environment where women believe their voices are heard. And I'm going to show a little video that the trainees in HSW 6 laboratories, this is a multi-lab effort. And last December for the ASCB Cell Biology Annual Meeting, which was held virtually, WICBI, Women in Cell Biology, put on a workshop on gender equity. And they asked me to put together some sort of a video. And so what I did actually is I just handed the people, trainees in HSW 6, a cartoon. And you'll see the cartoon at the end of the video. And it was a cartoon that Dean Reddy, who's sitting here, actually sent to me. So I think they just did a fabulous job.
All right. Thanks everybody for assembling here to plan the social activities for this year's retreat. Does anybody have any ideas?
How about a scavenger hunt?
Scavenger hunt seems like a lot of running around, right?
Well, I was thinking it could be good to get outside and get some fresh air. What if we did a scavenger hunt?
Oh, brilliant. Definitely. Let's do that. All right. Anybody have any other ideas?
I was thinking for an indoor activity, how about we do board games?
Board games. God, it seems like it takes too long. Anybody have any other suggestions?
Over here.
Yeah.
I was thinking we should do something indoors. So how about we do board games?
Oh, I love Monopoly. Let's do it. Any other ideas? Yeah.
What if we do trivia?
Trivia. That's a great idea, sweetie. Would any of the men like to suggest it?
Some of the men aren't smiling. Come on, Patrick, Okay, so in the last few minutes, and I'm going to buzz through this, let's talk science. And it, I guess as a sort of an umbrella, you could say that our research program aims to understand how cell behaviors are regulated and what goes wrong with disease. And notably, we do this across scales from molecules to cells to embryos to tissues to organisms. And we have focused over the years on two key regulators, actin filaments and architectures, as well as intracellular pH dynamics. And today I'm only going to focus on this second regulatory mechanism, which is intracellular pH dynamics. So when I first started my research program and started working on pH, sort of the going view or the prevailing view was that pH is rather constant for homeostasis, and that it might be dysregulated in some diseases. And through work from our group, as well as many groups throughout the world, our current view is that we now know that intracellular pH is indeed dynamic. In normal cells, it changes during the cell cycle with adhesion, migration, and differentiation, and that it is dysregulated in diseases. It's a hallmark of cancer that most cancers have a constitutively higher pH than a non-transformed cells. Many neurodegenerative disorders have a lower cytosolic pH than normal cells, and it's also dysregulated in other diseases. So my career, I kind of went back and went over and divided it into thirds. And in the very beginning of my career, we focused on how myriad cues, as shown here, regulate or cause a change in pH. And we showed how this is mediated by changes in the activity of different plasma membrane ion transport proteins, including acid extruders and acid loaders. And we went on to show that many of these transport proteins were actually substrates for kinases in canonical signaling pathways. And then I would say probably the middle of my career, we focused on identifying a number of different cell behaviors that were regulated by pH dynamics. And then in this third swan song of my career, we've really focused on the molecular mechanisms. And this is what we've done in collaboration with Matt Jacobson. So in terms of the behaviors, we predominantly focused on epithelial plasticity, where an increase in pH is now known to occur and be necessary for transformation, EMT, as well as differentiation of embryonic as well as adult stem cells. So for cancers, most cancers do indeed have a constitutively higher intracellular pH, as shown here. And I'm not going to go into the details on this, but over the years, we've shown how this enables cancer progression by allowing cell migration, directed cell migration, metabolic reprogramming, dysplasia, and I will be talking at the end on a project on charge-changing somatic mutations. In terms of stem cell differentiation, this is something we pioneered with Todd Neistel, and we were able to show that there was an increase in pH as naive mouse embryonic ground state stem cells differentiate and to the epiblast or state. And if we block that increase, we block differentiation. Todd went on to show, Todd's group, using these Drosophila lines that we had generated with the pH biosensor to show that during differentiation of follicle stem cells, shown here, to pre-follicle and follicle cells, there was an increase or pH gradient. And if we block that, we block differentiation. The flies are mostly infertile. We're continuing to understand, trying to understand at the molecular level how that occurs. And Yi Lu, a postdoc in the lab, has now shown using adult mouse intestinal stem cells that the LGR5 plus cells have a lower pH that are found in the crypt of these intestinal organoids, and that there's a pH gradient as cells differentiate. And if we block that increase in pH, we block differentiation. And what's interesting in this system, which the LGR5 plus stem cells are multipotent, we have, Yi has shown that it actually can regulate lineage specification. So there's different lineages, and an increase in pH is necessary for one lineage, but not another. And this is a whole new area that is just now being revealed. So how does this occur? And this is where I need to give a big shout out and thank you to Matt Jacobson, who together, I don't know, it was like together we got it in terms of, hey, something's going on here at the molecular level that people just aren't talking about and thinking about. And so we began to try to understand the design principles of what we call endogenous pH sensors. And we did this by bridging protein structure and electrostatics with cell biology, which really was kicking the soccer ball in a different direction. And we came to view this, and biochemists don't like this too much, but biophysicists embrace it. And that we've come to view protonation as a post-translational modification, very much like phosphorylation, acetylation, methylation, but it is much more difficult and much more challenging to study. It's not detected by mass spec. It's not detected by antibodies. It's not catalyzed by an enzyme. So it's a very challenging endeavor. Their specificity is achieved mostly by titratable side chains. This is mostly histidine, but we now know that pKa's can shift depending on the protein landscape, definitely for glutamic and aspartic acids. And as Christos, a postdoc in our lab, has shown, also for buried lysines. We've used a multidisciplinary approach to identify pH sensors. We always start with the cell biology and include a lot of biochemistry, recombinant proteins, structural biology. Matt has contributed molecular dynamics simulations. Mark Kelly, along the way, has helped with NMR analysis. We usually identify pH sensors. I can sort of divide it into three major ways. First is to start with the biology, and we've mostly always done that. But we have also been known to start with an intriguing protein structure or electrostatics. And as I'll show you in just a moment, also questions arising from disease charge changing mutations. So over the years, these are just some of the pH sensors that we've identified that have roles in cell polarity, adhesion, migration, tumorigenesis, metabolism, and proliferation. And shown in the red spheres are the titrating residues that we've identified. And it's important that these titrate within a physiological pH range of, say, 7 to 7.6, which is quite narrow within the cellular pH range. But notice something that's missing here is none of these pH sensors are direct regulators of gene expression. And we knew, especially through our work with stem cell differentiation, that pH was regulating gene expression. So we asked, how is it? And this is a very recent question we've maybe within the last, I don't know, Kyle, 2 years, 3 years, 2 years? How does pH dynamics regulate gene expression? And when we started to ask this, something very interesting was revealed, and that is that at least 65 different transcription factors have a conserved histidine in their DNA binding domain. And in all available structures, this histidine forms a hydrogen bond with nucleotides. Okay? And for some reason, this has just kind of escaped the notice in the field. So all members of the Fox family have this conserved histidine, all but 2 members of the Sox family, all members of the MIC, MITF, and TFE, TFEB families have this conserved histidine that in available structures directly forms a hydrogen bond with a nucleotide. Also, select members of the STAT and ETS family, and also ARNT, which is the HIF1 alpha subunit. So Kyle Kaiser, who's here, is a BMS student, began asking the question, does pH regulated protonation state of that conserved histidine confer DNA binding selectivity? So we're just beginning to investigate this question, but to sort of back up one step to explain the biophysical principles. So an adenine will always be a hydrogen bond donor. So when it binds to an amino acid in a transcription factor, that amino acid has to be a hydrogen bond acceptor. And if it's a histidine, it needs to be then deprotonated, say at a higher pH or neutral. In contrast, A thymine will always be a hydrogen bond acceptor, which means when it binds to an amino acid in a transcription factor, that amino acid has to be a hydrogen bond donor, which if it's a histidine would have to be protonated, say at a lower pH. So 2 examples, FOXN1 is shown in complex here with 2 different DNA motifs. Here it's shown binding to a thymine, and although this has not been reported, we would predict that that histidine has to be protonated. And when bound to a guanine, we would predict that this histidine would have to be deprotonated at, say, a higher pH. The same for MITF, which when bound to an E-box motif is bound to a guanine, which we predict the histidine would have to be protonated, versus when bound to an M-box and an adenine, the histidine would have to be deprotonated. This hasn't been published or shown. These are our predictions. Now, it's important that, a lot of things contribute to transcription factor DNA binding specificity. We're not saying that this is a binary switch, but rather could act as a coincidence regulator with cofactors, DNA architecture, all kinds of things that contribute. But so far, Kyle's data support our predictions. So here he's shown, he's made recombinant DNA binding domains of FOXM1 and FOXC2 bound to a DNA consensus motif FKH, which is shown here, and it would be binding to a thymine. So we would predict a higher binding affinity at a lower pH when the histidine is protonated. And that's precisely what he sees for both M1 and C2. He sees a higher binding affinity at 7 compared to 7.5 for both of these. And if he does a pH titration, he can show that binding affinity really titrates and is linear within the narrow physiological range of 7 to 7.6. And this just shows binding to a scrambled sequence to show specificity. So what about the significance of the histidine? Well, he's shown that in 2 ways. Number 1, by mutating to a lysine, which with a pKa of about 12 should always be protonated. We predict that he would see, and he does, a relatively high binding affinity, but that is pH insensitive. And he's also tested a histidine to asparagine mutation, which is one of the 3 most highly conserved mutations in C2 seen in cancers. And here you can see that binding affinity is markedly reduced, but it's also pH insensitive. So that really nails in the significance of that histidine for pH dependent binding. And notably, 2 other of these transcription factors have mutations in this histidine in the cosmic database. So is this real? I mean, what happens with recombinant proteins in a test tube is what can happen. It doesn't mean that it does happen. So we've now recently moved into cells, and with Francesca and Kyle, they have generated a fluorescent reporter assay, but just using this FKH motif to drive GFP. And if we express that alone in a cancer cell, these are triple negative breast cancer cells with a pH of about 7.7. There's very little GFP expression. If we co-express wild type C2, there's also very little expression. But if we lower pH with a pharmacological inhibitor of the sodium proton exchanger, we now markedly increase transcriptional activity of this or binding selectivity. So this suggests that this pH-dependent DNA binding selectivity also occurs in cells. Okay, I'm going to finish up real quickly here with this other question that Katie White, who's now an assistant professor at Notre Dame in the chemistry department, we started to ask about disease-associated mutations and specifically charge-changing ones. And the question we asked is, do some mutations generate a gain or a loss of pH sensing that occurs at the higher pH of a cancer cell? And might that be some sort of functional advantage? And one of the first things we noticed, and this was a collaboration with Matt Jacobson and also Ryan Hernandez, was that about 40% of somatic mutations in cancer encode a change in charge. This was rather surprising to us. So we started out with a very simple question, and that was arginine to histidine. So arginine with a pKa of over 12 should always be protonated, regardless of the pH. But now, if that is mutated to a histidine, that histidine could indeed titrate between the lower pH of a normal untransformed cell, in which it might be protonated, versus being deprotonated at the higher pH of a cancer cell. And one thing we also found initially is that arginine to histidines were enriched in cancer mutations compared to 1,000 genomes database, and that's shown here. And this is independent of codon bias and CPG site frequency. So I'm going to show you two examples that we've tested multiple, but these are probably two of the more common. One is the P53 ARG273 His mutation. This is one of the two most commonly occurring mutations in P53, and we know that P53 is one of the most commonly mutated genes in cancer. So this happens a lot. So here's our reasoning. In wild-type P53, that arginine, which is constitutively protonated, binds to the negatively charged phosphate backbone of DNA. Now, this is different than the transcription factor story I just told you, in which it was binding to a nucleotide. This is actually the phosphate backbone. Now, if this arginine is mutated to a histidine, at the lower pH of a normal cell, that histidine could be protonated, and you could still retain a little bit of binding and hence tumor suppressor activity of P53. Boom. Another mutation occurs, let's say RAS, pH increases, that histidine becomes neutral, and you lose DNA binding and P53 tumor suppressor activity. So Katie went on to show that. I'm just going to real quickly show you, we can show that in with recombinant protein and looking at DNA binding, that the wild type is pretty pH insensitive, but the mutant is exquisitely pH sensitive with much less binding at the higher pH of a cancer cell. We can look at transcriptional activity in cells. And here again, wild type protein, transcriptional activity is relatively pH insensitive, but for the mutant, it's exquisitely pH sensitive. And we can show the specificity for that histidine because an R to L substitution is pH insensitive. And then to look more globally, Katie did PCR arrays and identified a number of, as shown in red here, pH-dependent transcripts with the mutant, but not the wild type. And importantly, if we lowered pH, many of those transcripts with the mutant were now rescued to wild type levels. A second candidate recurring mutation we've looked at is the Egf receptor. And we've spent a lot of time talking to Natalia about that. And I want to just take 2 seconds to explain this. And that is in receptor tyrosine kinases, there is a segment, called the C helix, which when in swung out position, as shown in green here, the kinase is inactive. And the kinase is when the kinase is active in this cyan position, that C helix is swung in. Now, in the wild type receptor, this arginine forms a series of hydrogen bonds to stabilize that C helix in the swung out position or inactive. Now, if that's mutated to a histidine, if the histidine is protonated, you might retain some of that hydrostatic hydrogen bonding network. But if it's neutral, you don't. And one of the coolest things is when Diego, a graduate student at the time in Matt's lab, modeled this with molecular dynamics simulation and modeled the angle of the C helix to the center of the molecule. When that histidine is protonated, it adopted an angle very similar to the wild type arginine. However, when it was deprotonated, it now adopted a more swung in or a more narrow angle. Okay, and then Katie went on to show indeed that this oncogene had more activity at a higher pH, both with recombinant receptors shown here by looking at either autophosphorylation or substrate phosphorylation, also in EGF null cells, in which she expressed either the wild type or the mutant, and showed that even in the absence of ligand or in the presence, the receptor was more active at a higher pH, but the wild type receptor is not. And again, this was this histidine titration. The histidine is critical for this because an R2G was pH insensitive mutant. And then to look at a more complex behavior, she looked at colony formation or transformation in soft agar. Wild type EGF receptor was non-transforming. The mutant is transforming at lower pH, but it's more transforming at higher pH. And as a control, BRAF, which is pH insensitive, is transforming, but pH independent. So we've gone on, I'm going to kind of skip this to show this for a number of different arginine to his, as well as histidine to arginine mutations. But beyond cancer, what I want to emphasize is that there are charge changing mutations in many, many different diseases. And I think we've just touched the iceberg, tip of the iceberg on this. We have focused on charge changing mutations in cancer, but I think it's broadly applicable. This is just some examples, and there are actually hundreds of them. So, translational promise. Just a few slides. This is a translational award. I wouldn't call us a translational lab, but it's clearly always on our mind. And we used to think that targeting the increase in pH was a good way to go. And that was because we had shown a synthetic lethality. If we block proton extrusion with an oncogene, we could induce apoptosis. But since we started working on stem cells with TODs, and we saw that stem cells have a lower pH, and more recently we've shown pH heterogeneity in tumor cell organoids, that we believe these lower cells with a lower pH may actually represent the stem or cancer initiating cells. This idea of targeting the increase in pH is sort of we're not holding in favor as much as we used to. We're more now thinking about targeting the pH sensor itself. Now here's one example. I'm going to give you two. One is to design small molecule stabilizers, say that restore that mutant p53 binding to DNA at the higher pH. And then hence we can restore tumor suppressor activity. So we've actually begun doing this in collaboration with Emil Alexov at Clemson. They made a number of computational predictions of a small molecule that might restore this. This has a mind of its own. And from that, they selected a number of small molecules from databases, and they generated about 100 compounds that Kyle and Christos are now testing with recombinant p53, the GAD45 promoter, and fluorescence anisotropy. This is a sort of high risk, but really high gain project that we're quite excited about. Another way to target the pH sensor is to engineer a pH insensitive transcription factor. So I'll give you one example of one thing we've started testing, and that is with SOX4, the histidine 29 binds a thymine in the PUMA promoter. And this is going to induce an apoptosis as well as tumor suppressor cascade. And we predict that this is a high affinity binding at a lower pH. So now in a cancer cell, that's going to be turned off and have decreased binding. So the schematic is here, and this is what we're currently testing in an epithelial cell. SOX4 binds to this low pH consensus motif in the PUMA promoter to turn on or keep tumor suppressor activity on. Cancer cell now at a higher pH, SOX4 has a lower affinity binding to this promoter, and that attenuates any kind of tumor suppressor activity. But now if we express a histidine to a lysine mutation, we can rescue SOX4 binding and tumor suppressor activity. So these are questions we're currently testing. And I just want to end up with this. I've talked to you about how pH regulates cancer and stem cell differentiation, but I think it's important to know that it's broad. This whole concept of protein electrostatics and cell behaviors is very broadly applicable. In immunology, I think one of the most exciting examples is this VISTA reporter that has pH-dependent binding that's mediated at the lower extracellular pH in a tumor microenvironment. And this is a negative checkpoint regulator. So I know some companies are working on trying to develop ways to actually suppress this and rescue an immune response. Epigenetics, a number of writers and readers have been shown to be pH sensors. And in pathogenesis, we've known for a long time that a number of viral and bacterial proteins are pH sensors or pH sensitive. So again, this is an award that I share with everyone that has contributed to my absolutely amazing and thoroughly enjoyable career. So I thank you. Sorry.
Oh, Diane, I have something to say. Thank you for your great lecture. Yeah, I want to, on behalf of the academic senate, give you this small recognition. You can hang it on your
I will hang it
wall
on
and
my wall.
join your other awards that you've gotten over the years.
Thank you very much.
Yeah, thank you so much for doing it. So now we have some time for questions. So we're gonna have questions from the audience here, and then we'll also take turns with the audience on the Zoom. So yeah, who wants to ask? Holly. Oh yeah.
I too am suffering from a cold and not wearing a mask. I got COVID a month ago and it's nowhere like this cold. Okay. So just wear a mask, everyone. So it's so intriguing, the transcriptional changes, and you talked about it in the terms of cancer versus non. What organ or what cell in the body is going to use that dynamic information and do what it's supposed to do? What do you think?
The question is, where may this be relevant? And this is purely speculation. This is purely speculation, although we just got an NIH R21 grant with Dan Wagner to test it in zebrafish, is during development. And we know that these transcription factors are used reiteratively over and over again. They share a very similar binding, DNA binding domain, but they clearly regulate different. And it's not just on that FOXM1 is expressed here and FOXC2 is more expressed here. I think there's other layers of regulation. Now, I think it's possible, like in the SOX4 story, that it is indeed occurring in cancer, that where that higher pH is limiting a tumor suppressor activity. But I also strongly predict that this is happening during development. And that's why NeuralCrest gives us a perfect example to test it. Does that answer the question?
Hi, wonderful talk, Diane. I really enjoyed it. I'm trying to wrap my head around whichever the transcription factor was where the histidine is recognizing the thymidine, that site-specific recognition. Why does it still work if you switch it to lysine?
Because it's electrostatic.
Mm-hmm.
It's a hydrogen bond, and the lysine will be a hydrogen bond donor, and the thymine is a hydrogen bond acceptor, and the lysine with a pKa, unless it's buried, of 12 is not going to titrate. I don't think it's black and white, simple as that. Okay, I think that there are going to be a lot of nuances, but the fact that every single FOX transcription factor and all but 2 SOX have that conserved histidine, and in all available structures, that histidine forms a hydrogen bond with a nucleotide. And it blows my mind that it hasn't even been talked about. pH is just not on people's minds.
So Diane, I'm just I find it surprising that you see the elevated pH with differentiation and also in cancer cells.
Okay, well, this goes back to something that my lab talks about a lot, and that is cancer development gone wrong? One example is melanoma. So melanocytes differentiate from neural crest, right? And when they do that, I don't know, Cambria, you can maybe explain better since she's studying it, but melanocytes, so during the lineage specification, they turn on MITF, it's a transcription factor. And then when the melanocyte is fully differentiated, they turn off MITF. But in melanoma, MITF is now turned back on constitutively. And there is a school of thought that cancer is development gone wrong. It's not something you're going to ever test or prove, but I think, I don't know, Todd, what? Okay, so in the mouse embryonic stem cells, and we published this. This was the original J Cell Bio publication. Actually, what happens is that in the naive cell, pH is what, 7.2, 7.3, and then it goes up during the differentiation process. But by the time the epiblast cell is fully differentiated, it goes back down. So it's transient. Does that? But if it stays up, then to me, that's cancer. Does that?
Questions on Zoom? Penny?
Hi. I have a question. Is pH dysregulation associated with pain?
With what?
Pain.
With pain.
Pain.
Yeah.
Alan, you want to comment on that? Well, I mean, trip channels are pH sensors, are pH sensitive. Yeah. Not something we've studied, but.
Can you guys hear from that microphone? Can people on Zoom hear with the other microphone as well? No? Did you guys hear the answer? Maybe you have to use this microphone. I don't know if they hear it. Yeah, you might have to use that microphone. Yeah, let me get this just for the audio. Yeah. Let me just take this to the audio.
Who had a question? Kevin?
You mentioned that there's two SOX proteins that don't have that conserved histidine.
In SOX, in the SOX family?
Yeah.
Yeah.
Have you looked at those two?
No,
Are they?
those are going to be obviously good controls.
I wonder if is there anything interesting about their transcriptional profile, or maybe they're expressed in cells that have
You know, we
different
haven't gotten
pHs?
into it. We've mostly focused on the FOX so far, but I mean, we've talked about the two SOX and have been working on the SOX4 story with reactivating a tumor suppressor pathway. But we haven't, Kyle, we haven't really talked about that.
Questions on Zoom?
Yes. I have a question from Catherine Streetinger. Awesome and inspirational talk. Thank you. My question is, do you have an in vivo evidence of pH-dependent stem cell differentiation?
Can someone reinterpret that question for me?
I think that's
in vivo, you know, that's a real challenge.
It's got quotation marks around it, in vivo.
In vivo, that's why we have used more model organisms. We've used the Drosophila eye epithelium to show pH changes in real time in a intact animal, and we're starting to do this with zebrafish. This is not something we could readily do with mice. I'm sure with some sort of fancy imaging modality someone could try. It's not something we've attempted. You know, we've used the Drosophila eye epithelium as an external organ that we can view, and it's turned out very interesting for us. The intestinal mouse intestinal were
those are
organoids.
quite good at differentiating in.
They
into
were
all the
intestinal
different
organoids.
cells. Yeah.
And you can show the raw data expressed a pH biosensor and could show in the organoids that pH is lower in the crypt and higher in the differentiated villus cells. And if you block that pH gradient, you block differentiation, particularly to the secretory lineage, but not the absorptive lineage. So that's what I was implicating in terms of lineage specification. So you completely block ATOH and the secretory lineage, Panus cells and tough cells, but not the absorptive lineage of enterocytes.
Diane, as I said before in my introduction to you, talking about the nerves, external regulators of pH, right? And so you've got synaptic connections with many basal progenitor cells, epithelial progenitor cells that may orchestrate changes, intracellular pH in that way. I'm saying that because a lot of, adrenaline causes cyclic AMP enrichment and a whole bunch of other processes that might be involved with changing and altering that internal pH. So have you looked at that and thinking about the external contributors, not just the regulators that,
Yeah, we haven't too much. I mean, we've looked at a couple of processes in which they're transient changes. For example, during the embryonic stem cell differentiation, it's transient. It goes up and then it goes down. Cell cycle. So we could show this, we did this years ago, that there's a transient increase in pH right at the end of S phase. Yeah. And if you block that increase, you delay G2M. You don't block it. It's not obligatory, but it's permissive.
Yeah.
So you delay G2M if you block that increase in pH.
Yeah. I suppose I'm really interested in the niche environment and potential changes that are associated with that niche, because the niche changes obviously during development immensely into an adult stage, and then you get aging, which I'm also intrigued at to see if there's an alteration in pH along that because of essentially the dysplasia that occurs with aging. So just a couple of thoughts there on external stimulators.
Open field in terms of pH and
Right.
development. Virtually, I mean, poor Cambria is really sort of writing a new book on it, and for her quals and her proposal, they're just was there like 2 papers and it's an open field for someone who wants to.
Yeah, no, it's cool.
Go down that direction.
Any more questions?
With Todd, and we've really shown that it's clearly important in stem cell differentiation.
Right. Yeah, absolutely.
I'm convinced. I think you are. And a couple of other instances have shown it, but not broadly. I mean, pH has never been sexy.
It's hard. That's why.
It's not. I don't say that with bitterness. It's true.
Yeah, salivary glands have never been that sexy either, but someone's got to do it, Thank you. Zoom, any more questions via Zoom? We're done?
Oh, actually, before.
She's pooped, everybody.
Okay.
You've got any more questions?
Good, no more questions, but I do have an announcement so there will be a reception outside just outside the auditorium for Diane, and everyone's welcome.
Thank you, everybody.
Oh,